Hello biology students, and welcome to video 2 of the genetics unit. So here we're going to go back and look at those same crosses that Mendel did with the pea plants that we saw in the last video, only this time we're going to be getting a little bit more mathematical and looking at why he saw the traits that he did in various generations. So just as a reminder, remember that when Mendel crossed a true breeding purple plant, so in the parental generation, he had a plant that was true breeding purple, so capital P, and he crossed it with a true breeding white plant that was little p. Remember, his F1 generation was all purple, and we know, now that we know some stuff about alleles and genes, that his F1 was all capital P, lowercase p, so they were all heterozygous for the color gene. So if we kind of ask, well, did the white trait disappear? Well, not really, because these heterozygous purple plants were still carrying that little p recessive allele. So when he let the F1s, so these heterozygous plants, self-pollinate, he saw in his F2 that his plants were 75% purple and 25% white. So the white trait had reappeared. So in this video, we're going to be trying to figure out, well, why was he seeing this 75 to 25% ratio? So one of Mendel's first big discoveries was what he calls the law of segregation. Basically, the white allele, the allele for the white flower, is segregated or separated, traveled separately from the purple allele. So let's look at what this means on a chromosomal level. So we know that chromosomes come in homologous pairs, and those homologous pairs have the same genes, but that they have different alleles. So therefore, because in gametes, in sex cells, we only have one copy of each homologous chromosome, that means each gamete, each egg or sperm, only carries one allele for each inherited character, or for each gene. So maybe if this is a homologous pair, and I'm making sperm cells, one sperm cell is only going to get one allele. So even though the diploid cell we started with might have had a capital B and a lowercase b, these are going to segregate. They're going to separate during gamete production. So to kind of reiterate this point a little bit, so diploid organisms have pairs of homologs, right? And those homologous pairs have the same genes, but because they have two chromosomes, there are two copies of each genes. So when we're making egg or making sperm, those chromosomes separate, and each new gametic cell only gets one copy. Okay, so let's look at this process. So here I have a diploid parent, and we're going to go through meiosis. So we're going to go through S phase, and we're going to copy that chromosome. But then look at the two alleles, my capital S and my lowercase s, are segregating or separating. So now some of these sperm cells carry the capital S allele and some carry the lowercase s allele. So this was Mendel's law of segregation, basically the idea that the two copies can travel separately from one another. And therefore gametes only have one allele for each gene. So this is not really a big deal if you're homozygous, because then all of your sperm or eggs are going to end up with the same allele because you only have one type of allele. But this does matter when you're heterozygous, because if I'm heterozygous and I'm making egg cells in my body, I can either donate a dominant allele, or I can donate a recessive allele to my offspring. Okay, so let's look at a cross mathematically. So here I have the parental generation, and this kind of copied weird onto my video making software. So this capital T, and lowercase t should be over here. So here it's kind of asking, okay, what generation do we have? And if we're looking at the first generation, the answer is the parental generation, okay? So this dad and this mom are the parents. And the question is, well, what gametes can they produce? So if these parents undergo meiosis, so this dad makes sperm cells, he would either donate a capital T or a capital T, and the mother would donate either a lowercase t or a lowercase t. So now, if we want to figure out what alleles are the offspring going to have, we can fill out what's called a Punnett square. So I'm going to show you a Punnett square right over here. So what we're going to do, we're going to take the alleles for one parent, and we're going to put them on one side of the square, and take the alleles for the other parent and put them on the other side of the square. So that means, if an egg with a capital T, a sperm with a capital T combines with an egg from a lowercase t, that offspring would be heterozygous. Same thing here, if this sperm and this egg combined, I would get this heterozygote. If this egg and this sperm combined, I would get this. And if this and this combined, I would get this. So you can see in this case, all of the offspring would end up being heterozygous. So you can see that over here. Okay, so you can see that we can say all of the genotypes are going to be 100% capital T, lowercase t. And in this case, the phenotypic ratios, 100% of the offspring are going to be tall because the capital T is the allele for tall. So let's go back to Mendel for a second. So we were just looking at tall and short plants. Let's relate this back to the purple and white flowers that Mendel was looking at. So what we just looked at in our Punnett square was the equivalent of this part of this chart. So we had a homozygous dominant parent and a homozygous recessive parent, and we found out that 100% of the offspring were going to be heterozygous. So they were going to show, in this case, the purple trait. So now what we're going to look at in the next slide, we're going to do a Punnett square for crossing two F1 plants together. So we're going to cross two heterozygous plants and try to figure out why we end up with a 3-to-1 ratio like we saw at the very beginning of the video. So you can see here are my two F1 plants, my two heterozygotes, and if I cross these together, now the dad could either produce a capital T or a lowercase t, and the mother could also produce a capital or a lowercase t because they're both heterozygous. So if I put, these gametes on the outside of the square, you could see that if this gamete and this gamete combine, I have a homozygous dominant. If this one and this one combine, I get a heterozygote. If this one and this one combine, I get a heterozygote. And if this one and this one combine, I get a homozygous recessive. So you can see my genotypic ratios, my ratio of genotypes is 1 capital to 2 heterozygous to 1 lowercase. And this, if I calculate my phenotypic ratio, which is what that stands for, 3 of those plants are tall and 1 is short. So that's how we got that 75 to 25% ratio, that 3-to-1. Okay, so it turns out that we can also explain all of these ratios using probability. And we're going to talk about probability now, and it's not going to come back for quite a bit later in the unit, but it's kind of interesting to talk about it now while we're still dealing with some simple genetics. So what probability is, it's the likelihood that a specific event will occur. And you can calculate probability of the number of events times you expect an event to happen over the number of times an event could happen. So for example, if we were calculating the probability of picking a heart in a deck of cards, well, you would expect this to happen. There are 13 heart cards in a deck of cards out of 52, so that would reduce down to 1/4. If you're calculating the probability of picking a king out of a deck of cards, well, there are 4 kings. Let me erase my previous math here. There are 4 kings out of 52 cards in a deck, so that would reduce down to 1/13. If I wanted to know the probability of picking the king of hearts, I could multiply those two probabilities together and get 1/52. Or the probability of not picking the king of hearts, well, it would be every card, so there's 51 non-king of hearts cards out of 52 total. So those are just... So a couple other things about probability that I want to talk about. The probability scale runs from 0 to 1. That means a probability of 0 means it's definitely not going to happen, and a probability of 1 means an event definitely will happen. So if you're ever calculating probabilities and you get something below 0 or over 1, you're not doing it right. There's something wrong. Okay? And also, the probabilities of all possible outcomes must add up to 1. So for example, if you're tossing a coin, you have a 1 out of 2 possibility of tossing a head, and a 1 out of 2 possibility of tossing a tails. So those two things, they have to add up to 1 total, or again, something's not quite right. So there are 2 main rules that we're going to look at in probability to help us explain genetics. The first one is called the rule of multiplication. Basically, the rule of multiplication states that if you have independent events, so if you have events that do not affect one another, you can multiply their probabilities together to determine the probability of both things happening. So for example, if you toss a coin in the air, the probability that you toss heads the first time, like that you get heads, is not affected by any other coin tosses you did in the past. So if you got 49 heads in a row, that does not affect what your 50th toss is going to be. They're completely independent. So if I want to calculate the chance that two coins are tossed simultaneously and they're both heads, I can multiply the probabilities together because the two coins don't affect one another. So there's a 1/2 chance that the first coin will be heads, and there's a 1/2 chance that the second coin will be heads, and I can multiply these together and calculate that there's a 1/4 chance of them both being heads, and I can multiply them because they do not affect each other. So let's apply this to genetics now, okay? So inheritance, it turns out, follows the rules of multiplication, okay? So basically, there is so if I have two parents and they're both heterozygous, so I have a heterozygous female and a heterozygous male, there is a 1/2 chance that this male will donate a big B, and there is a 1/2 chance that the female will donate a big B. And because what the male donates is not affected by what the female donates, so they're independent events, I can multiply them together and get there's a 1/4 chance of the offspring being capital B, capital B. Okay? So we can do that for each of the probabilities here. So if I have a question, two F1 heterozygotes mate, and what is the probability of having a homozygous dominant child? Well, that's the example that we just did. So if I have these two parents, there is a 1/2 chance that this parent will donate a big B, and there is a 1/2 chance that this parent will donate a big B. So I can multiply them together and get that the chance of the child being big B is 1/4. So the other rule that we're going to look at is the rule of addition, and this one's a little bit more confusing. And the rule of addition says that if there are two ways of getting the same outcome, you can add the probabilities together to figure out what the total probability is. So for example, if I have, again, two heterozygotes parents, and I want to know the probability of the child being heterozygous. Okay, so there are two ways this could happen. This parent could donate a big B, and this parent could donate a little b. So that's one way this could happen. Or this parent could donate a little b, and this parent could donate a big B. So there are two ways to get to the same outcome. So therefore, I can calculate the probability of this happening, and I can calculate the probability of this happening, and if I add them together, I'll get the total probability or the total chance of getting a heterozygous offspring. Okay. So let's do just that. So let's calculate the probability, oops, didn't mean to move that. Let's calculate the probability of this first combination first. So I'll write it down here. So here are my two parents again. Okay. And I know that, so for this one right here, I know that this parent has to give a big B. So there's a 1/2 chance of that happening. And there's a 1/2 chance of this parent right here giving a little b. So because these are independent events, I'm going to multiply them. So there's a 1/4 chance that I'm going to have that offspring. So now let's calculate the probability of this one happening. So there's a 1/2 chance of this first parent right here giving a little b. And there's a 1/2 chance of this parent giving a big B. So if I multiply those together, I get 1/4 chance of little b, big B. Now because these are two ways to get to the same outcome, I can add them together using the rule of addition, and I get a total 1/4 times 1/4, that's 2/4, or 1/2 chance of having a heterozygous offspring. Okay, so that's it for video 2. I'll see y'all in video 3.